thank you, Jerome, for that kind introduction. And I was actually actively involved in helping to try to recruit Jerome and some of his colleagues here, and I was ecstatic when we were successful. And I think probably the entire community has grown to appreciate his value to the whole cardiac field here at Washington University, and he's playing an important role in bridging the relationship between the medical school and this campus. So today what I'm going to talk to you about is our efforts to understand the diversity of potassium channels. And in particular, I'm going to talk about voltage-gated potassium channels and try to make a few analogies to these channels and other types of channels, potassium channels in the myocardium. So I'll start by telling you sort of the big picture of why we're interested in this problem. And then I'm going to move on to what we, to sort of summarize in a couple of slides, a lot of years' work characterizing the physiology and the biophysics of these channels, and then move to the molecular basis of this diversity. And then I'm going to end up with some of our more recent work focusing on trying to understand the mechanisms involved in the modeling of these channels in the myocardium, both under physiological conditions but also under pathophysiological conditions. So as the mantra goes in my lab, voltage-gated potassium channels are the most important channels. So if you remember anything that I say today, that would be the one thing that you should remember. That they are more important than other channels in shaping the electrical activities, not only in myocardial cells, but in all sorts of other excitable cells, as well as in what are classically considered non-excitable cells. And so if you were to be able to put an electrode into different regions of the mammalian heart, and this is a schematic of the human heart, you would find that the waveforms of the action potentials that you measure are really very distinct. And many years ago, we and other people recognized that one of the biggest differences, particularly if you focus on the ventricular myocardium, was the durations of the action potential and the rate at which the action potential repolarizes. And so we and other people, probably 15 years or so ago now, speculated that these differences could be due either to differences in the types of channels that are expressed in different myocardial cells, or simply due to differences in the densities of the same types of channels. And so using a variety of electrophysiological techniques, people have identified a whole host of different types of channels that underlie action potential repolarization in the myocardium. So I just want you to focus on the lower half of the slide, where we're focusing on the potassium channel. So there are two types of transient outward current channels, called transient because they activate rapidly and inactivate rapidly. And then there are a host of delayed rectifier type currents, IKR, IKS, and then these others, I steady state and IK slow, which I'm going to come back to in a bit. But these channels, the delayed rectifiers, activate more slowly after the onset of depolarization, and they contribute to determining both the shape and the duration of the plateau phase, and also the details of this repolarization phase. So these descriptions of these currents were initially based on electrophysiological tools to attempt to separate the different current components based on their time and voltage-dependent properties. And subsequently, people moved in to use a variety of different pharmacological tools and could actually show that these currents actually had different pharmacological properties. And that led people to the suggestion that they probably were encoded by distinct genes. And I'm going to tell you about some efforts to figure out that. So in large mammals, largely through the work of Charlie Anselovich and his colleagues, it was recognized that the expression of different voltage-gated potassium channels are highly variable through the thickness of the ventricular wall. And we, because we wanted to use molecular genetics in mice to be able to dissect the individual current components, decided to ask the question of whether or not in mouse you see this same heterogeneity in the voltage-gated potassium channel currents. And importantly, you do. So these are recordings. These are voltage clamp recordings under conditions in which we've isolated only the potassium currents. So these are currents activated only by depolarization. They're not dependent on intracellular calcium influx. So they're voltage-gated. They're not calcium or dependent on any other known intracellular second messenger. So these are recordings from right ventricular myocytes and from cells from the apex of the left ventricle And I think what you can see in both cases is you have rapidly activating currents, that a component of the current inactivates very rapidly, and then there's a slowly inactivating component, and then what we call the steady state current, which is basically a non-inactivating current. No matter how long this voltage step is, this current will continue to be present. So if you just compare these records in the right ventricle and the apex, the main difference, and it turns out quantitatively to be the case, is in the expression level of this fast transient outward current. The currents are highest in the right ventricle, lower in the apex of the left ventricle. They're lower still in the base of the left ventricle in records that I want to show you. But then also in the interventricular septum, the current recordings are actually even more distinct. So there are some cells in the interventricular septum that also have a fast transient current, and there are also cells that have what we call the slow transient current, similar to what I illustrated in the previous cartoon. And then there are other cells in the septum that lack the fast transient current but do express this slow transient. So obviously, these recordings don't come out of our amplifier all pre-labeled like this, so what I'm summarizing is a lot of results of a lot of experiments, and I'm going to go on to the molecular dissection, so you can argue about whether these descriptions are valid, but I hope that I'll be able to convince you with the molecular analysis that they are. So there's several important things that came out of these. One is that these currents are quite distinct in terms of the rates of inactivation. So the fast transient currents inactivate very rapidly, fast time constant. The slow transient current, the inactivation is about 5 or 6-fold slower. These are these rates here. And then the slow delayed rectifier type currents are much slower to inactivate. The properties of these currents are identical throughout the ventricular myocardium. So the kinetics of ITO fast in the right ventricle, in the apex at the base of the left ventricle, and in the interventricular septum where it is expressed are indistinguishable. In addition to inactivating with different time courses, these currents also recover from inactivation with markedly different kinetics. So the fast transient current, which inactivates rapidly, also recovers from inactivation very rapidly. And both the slow transient current and the delayed rectifier current recover slowly from inactivation. Another argument that these are in fact distinct molecular entities that encode these different channels. Okay, so what do we know about the molecular basis of the diversity of potassium channels? Well, this is a schematic of a core-forming subunit, an alpha subunit of a voltage-gated channel. The first one was cloned in the late 1980s from Shaker locus in Drosophila, and predicted a 6-transmembrane spanning protein with a highly charged S4 region that was speculated to be the primary voltage sensing region of the channel. These are channels that are opened only by changing the membrane voltage. And then a region between the 5th and 6th transmembrane domain that was thought to dip back into the membrane to contribute to the potassium selective pool. If you take cRNA from this cloned subunit and express it in the heterologous expression system, the early experiments were done with xenophocytes, you get rapidly activating, rapidly inactivating currents that are only dependent on changes in membrane voltage. So they're voltage-gated currents, not dependent, again, on calcium. So in Drosophila, it was immediately recognized that there was a considerable extent of alternative splicing of the Shaker transcript. So there's a lot of splicing in both the N-terminus and the C-terminus. And interestingly, if you express the different splice variants in a heterologous expression system, you produce voltage-gated potassium channel currents with markedly different kinetic properties. So this is potentially the first way to get functional potassium channel diversity through splicing of single gene products. And it was speculated at the time, and now it has been proven, demonstrated, I think beyond the shadow of a doubt, that these alpha subunits co-assemble as tetramers to generate potassium selective channels. So the cloning, the identification of the Shaker locus put a very large number of laboratories to work for about the next 5 years into the early 1990s, looking for first other Drosophila homologs of the Shaker locus, and then expanding into vertebrate systems. And this resulted in the identification of about 25 different potassium channel alpha subunit genes in vertebrates. And they were then grouped by families based on sequence homologies. So the Shaker is the KB1 subfamily, so that's potassium channel voltage-gated subfamily 1, and then each of the other subfamilies were grouped based on sequence homology. In Drosophila, there's only 4 families, KB1, 2, 3, and 4, sorry. And then in the vertebrates, there's a host of other different subfamilies. And within each of these families in vertebrates, there are many different members. So in the KB1 subfamily, there's 11 different members. The KB2, there's only 2 different members. KB4, there's 3. So now I think if you try to think in terms of the map, if you could potentially mix and match these subunits coupled with alternative splicing, you could potentially generate many different, functionally different potassium channels. There are some rules, and these have not been demonstrated in real cells, but in heterologous expression systems, there are rules for how these subunits can combine. So individual subunits of the KB1 subfamily, for example, can combine with one another, but they don't co-assemble with members of the KB2 family. So there are some rules, and that's linked to specific sequences primarily, but not exclusively in the N-term. So in addition to these core-forming subunits, there have now been identified a whole host of accessory subunits in this channel. Most of these were identified using yeast-2 hybrid approaches, and in a couple of cases using biochemical methods of immunoprecipitation. And the first of these are the MIN-K, the minimal potassium channel subunit subfamily, which got its name based on the fact that when it was first cloned and expressed in xenophysiocytes, this subunit all by itself made functional voltage-gated potassium channels. And so people thought that you could get a potassium channel from this little single transmembrane domain expanding protein. It was later shown that xenophysiocytes express an alpha subunit endogenously with which this accessory subunit combined to give functional channels. That actually illustrates probably one of the biggest problems with using heterologous expression systems to study functional channels if you want to know how they really work in real cells. Because in most expression systems, you simply don't know what other accessory subunits are present. There are also intracellular accessory beta subunits, which were first identified also in Drosophila and subsequently in the mammalian brain. And they were identified as binding partners of the KB1 family of alpha subunits. They are related to oxidoreductase enzymes, and they have an NADPH binding site. And interestingly, when the beta subunit is crystallized, it crystallizes with NADPH bound to this site. So there's a lot of speculation as to whether these beta subunits may play a role in regulating the redox state of channels in intact cells. And then there are potassium channel interacting proteins, the K-chips, which are related to the neuronal calcium sensing proteins, which have multiple EF hand domains or calcium binding domains. And these were originally identified as specific accessory subunits of the KB4 family. There are many members of this family. There's alternative splicing of all these transcripts. The same is true for the beta subunits, and the same is true for the MIN-K subunits. And then there are 2 additional accessory subunits, another one that's intracellular, which is called potassium channel accessory protein, and another protein, which is also a transmembrane protein like the MIN-K protein, which was identified in brain, and this is related to the aminopeptidyl transferase family of enzymes, but doesn't seem to have any enzymatic activity. At the moment, the role or even the expression of this subunit in the heart is a matter of speculation. But So now we can see that we have ever increasing complexity as to how functional channels might, in theory, be made up in cells. And so the question becomes, how do we figure out the relationship between all of these subunits and the channels that are expressed in cardiac myocytes, particularly ventricular myocytes. So as you know, and this is just a reminder because this is probably the best example of a case where molecular genetics has really provided insights into the diversity of these channels. So there are two long QT syndromes that are linked to mutations in two voltage-gated potassium channel forming alpha subunits. The first is long QT1 linked to mutations in KCNQ1 or KBLQT1. And importantly, most of these are missense mutations, and interestingly, the mutations are found throughout the protein sequence. And most of them are dominant negative effects resulting in reduced expression or lack of expression of functional channels of the subsurface. The others are mutations in the ERG1 or the KCNH2 locus, and these are linked to long QT2. And importantly, these mutations are also found throughout the protein sequence. There are a variety of them. There's frame shift, missense mutations, deletions. Many of them are dominant negative, but in addition, some of the mutations also result in trafficking defects. More recently, mutations in both of these genes have been linked to congenital short QT syndromes, which are also inherited arrhythmia syndrome. And recently, mutations in KCNQ1 have also been linked to atrial fibrillation, so another congenital arrhythmia. And I think the study of these genes probably is one of the best examples of where people have made great efforts and now great strides in linking genotype to phenotype in patients, which actually has led to improved clinical treatment of people whose inherited. They are very rare, however, and so it's still important to study the mechanisms of arrhythmia in people that the arrhythmias are not congenital, and certainly understanding the functioning of these proteins as well as all the accessory subunits and other regulatory proteins that they might interact with are probably going to be important. So we have focused our efforts largely on understanding these transient outward current channels, and we've, as I said a few moments ago, to do this we've actually focused on using the mouse. But it's important to say up front that a mouse is not a human. It's not just a scaled down, smaller version of a human with a tail. It actually is extremely different in terms of cardiac physiology if you look at both the heartbeat and the waveforms of individual action potentials in the mouse myocardium. So the normal resting heartbeat of a mouse is about 750 beats per minute. So it's about 10 times faster than a human heart. That means that the action potentials have to be much briefer in order to be able to sustain those high frequencies. These are typical waveforms of action potentials recorded from mouse ventricular myocytes. So they really look very different. And for those of you who are really have grown to love these action potentials with these very pronounced plateaus, you can see that this is completely lacking in the mouse. But the good news is that if we look at the ionic currents that are expressed in these cells and ask the question, do they look like the currents that are expressed in larger mammals, the answer is yes. So here, if we just focus on the transient current, there's a fast transient current, as I showed you in the recordings of myocytes. It turns out that the properties of those channels are very much like the fast transient current channels in humans and other large mammals. And then similarly, there's a slow transient current, and it's very similar to the slow transient current in large mammals. The delayed rectifier currents, in contrast, are quite different. So we've identified three novel components of delayed rectification in the mouse ventricle, which we've termed IK slow 1, 2, and I steady state. And these are either absent or probably not very important in larger mammals. The genes, the KCNQ1 and KCNH2 that encode the delayed rectifiers in human are present in the mouse. They are expressed, but those currents are not functional during these action potentials because the action potentials are so brief, too brief for there to be any activation. So what I'm going to do now is to focus my talk on sort of our efforts to understand the molecular basis of of these to transient outward channels. I didn't say it at the beginning, but please feel free to interrupt me if you want to ask a question directly. Okay, so just to come back to the phylogenetic tree of these alpha subunits, for a variety of reasons, which I'm not going to elaborate in detail, we speculated that fast transient outward currents would be encoded by members of the KB4 family, and that the slow transient currents would be encoded by members of the KB1 family. And this is really based on expression levels. It's based on what the channels look like if you express them in heterologous expression systems. But what we set out to do was to develop a dominant negative strategy that would basically allow us to probe the role of each of these individual KB alpha subunit families in the generation of channels in intact cells. So we made dominant negative constructs of KB4. And the way we did that, actually, well, initially we did it by a variety of different methods, but it turns out, in hindsight, the best method to use, and the one that we now use exclusively, is to introduce 4 mutations into the alpha subunit. And the idea behind this is to generate a protein that is as close to being the wild-type subunit as possible. And in this case, in the case of the KB4, we generated a single point mutation in the 4 region that resulted in a subunit that, when expressed, didn't produce any functional multiplicating chain. So for it to act as a dominant negative, you would like that when you co-express this with the wild-type subunits, that you selectively attenuate channel currents encoded by KB4 subfamily members. So these are currents that are produced when you express KB4-2, and these are in heterologous expression systems now. So this is 4-2 expressed by itself, and then 4-2 with the dominant negative, and KB4-3, another member of the KB4 family, also expressed with the KB4-2 dominant negative. So in both cases, you get either elimination or marked reduction of the current. And the only reason I show you this one example where there is some current remaining is to remind myself to tell you that when there are residual currents present, those currents have properties identical to the wild-type current. So that means simply that those are currents that are not contaminated by the presence of the dominant negative construct. And if you do co-expression studies and explore the stoichiometry, you find that it only takes one mutant subunit to poison the channel.
Is this because traffic? Do you know if it's for trafficking reasons or?
No, as far as we can tell, these subunits co-assemble with the wild-type subunits, and they traffic to the cell surface, and they just
they are in the surface, but they are not conducted.
That's right. They're up. Pardon? That's right. Exactly. No, they're not. These are not GYG, actually. Actually, what we did here was we used an approach that was developed by Pancho Bensanilla to study gating currents of potassium channels a number of years ago, and he made mutations in the Shaker locus and showed that this one, it's a phenylalanine to tryptophan mutation that's not in the GYG. I have illustrated GYG here. That's another way you can do it, but this one example we didn't do that.
What's the ratio of the genes that you express so you can suppress all
Here,
the current?
these examples, I don't remember. They could be 1 to 1 or so.
1 to 1,
Yeah.
and you don't see the current.
And then, importantly, if you express this KB4 dominant negative with other KB alpha subunit subfamilies, you don't get dominant negative suppression. So that's really important because we wanted to use this approach in vivo. And we wanted to be able to say that the currents that are affected by expression of the dominant negative reflect the channels that are encoded by genes of that subfamily. So what we did was we took this mutant, we put an epitope tag on it so we'd be able to identify it in the transgenic mice, and then we put a cardiac-specific promoter in front of it and expressed and generated mice expressing this construct. So I'm just going to show you sort of the summary records of what we found from these experiments. So these are recordings, again, similar to the ones that I introduced you to a while back. These are recordings from wild-type left ventricular apex of the interventricular septum, and showing that, again, this very large fast transient current in the apex but not in the septum. And then if we look at myocytes from the transgenic animals, I think you can immediately appreciate that this fast transient RV current is gone. It's not necessarily true that you would have predicted that you would eliminate the current totally, but it turns out that when we make transgenic animals using this sort of approach, you have two things. One is you have multiple copies of the transgene that are actually inserted, and in addition, this particular line of mice, it was 5. And then in addition, it's a very strong promoter, so this mutant protein is abundantly expressed. Actually, that turned out to be lucky because it made analysis and interpretation of the results much easier. But then interestingly, it's hard to see here, but I think I'll be able to show you the next 2 slides. When we analyzed these records from the apex of the dominant negative myocytes, we found the presence of a slow transient current identical to what we had seen in the septum cell. So we weren't really sure whether or not eliminating the fast transient current simply revealed that current, or whether there was an upregulation of that current as a result of the loss of the fast transient current. So there was remodeling, if you will, of the potassium currents as a result of losing or eliminating one. And one of the sort of signatures of this finding was that in the dominant negative myocyte, what we found was that the rate of recovery of the peak current from inactivation was very slow, with a time constant very similar to what we saw for the slow transient current in the septum cell. And this is a fast recovery for ITO fast and wild-type cells. And as I indicated a couple slides back, we had speculated for a variety of reasons that the slow transient current would be encoded by a distinct gene family, and specifically by the KB1. And at the time we were doing these experiments, Barry London and his colleagues made a knockout of the KB1.4 locus, so we speculated, or we thought it would be worth testing whether or not KB1.4 could encode the slow transient current. So these are Western blots of ventricular membrane proteins from wild-type and transgenic animals. And just focus your attention here, there is an upregulation in these animals of the KB1.4 protein. A kind of dirty Western blot. So what we decided to do then was 2 separate experiments. One is to ask the question, what is the role of KB1.4 in the generation of the endogenous ITO slow in the septum? And then secondly, in the dominant negative background, does the elimination of 1.4 eliminate that slow transient current in both the septum and in the apex where the slow transient current is not normally expressed? So these are recordings from the 1.4 knockout myocytes, again from the apex and the septum. It's a little bit difficult to see, but I think you'll be able to see it more clearly in a moment. But the slow transient current, when you analyze these records, is gone from the septum. There's no detectable effect of the knockout of 1.4 in the apex. And these are the records you saw before from the dominant negative. And now when we express the dominant negative in the 1.4 knockout background, I think you can see now that we completely collapsed the gradient of outward current expression through the ventricle. So we've now eliminated the fast transient current and the slow transient current, both in the septum and in the apex. So that's actually quite interesting because it tells you that the remodeling of the potassium currents is a highly selective process. When we eliminate ITO fast, you get an upregulation of ITO slow. But if you eliminate both of them, you don't get any further remodeling. Okay, so what accessory subunits might contribute to the formation of these channels? So the K-chip, as I mentioned, were originally identified in a yeast 2 hybrid screen by binding to the KB4 N-terminus. And the KB beta subunits were originally identified based on immunoprecipitation studies from brain as being specific for KB1 subunits. So what we decided to do was ask the question, is K-chip expressed in the mouse heart? Is it associated with KB4 subunits? And does it play a role in the generation of the fast transient current And for the beta subunits, the same sort of question, but now instead of for the KB4 encoded currents, for the KB1 encoded currents. So we used biochemical methods actually to probe the interaction between the K-chip protein and the KB4 proteins. In the mouse, there are two KB4 alpha subunits, 4.2 and 4.3 expressed. And we could show in co-immunoprecipitation experiments that these two subunits co-immunoprecipitated, which suggested that they both contribute to the formation of these fast transient outward current channels. So the channels are heteromeric, if you will, they're not homomeric. And in these experiments, what we wanted to ask was whether or not K-chip also co-assembled with the KB4 subunit. So we did two experiments, immunoprecipitation with the K-chip antibody and immunoprecipitation with the KB4 antibody, and then probed with the alternate antibodies in both cases. So when we immunoprecipitate with the K-chip and probe with 4.2 and 4.3, you can see both 4.2 and 4.3 co-immunoprecipitate with the K-chip. In the reverse experiment, precipitating with 4.3 and probing with K-chip 2, I think you can see that the K-chip 2 co-assembles with 4.3. And interestingly, if we look at the supernate in these immunoprecipitations, there's very little K-chip 2 left, suggesting that the KB4 alpha subunits are the primary partners of the KB4 alpha subunits in the mouse microorganism. So one of the interesting things that we wanted to know was what underlies this gradient of expression of the fast transient outward current in the ventricle. And so we asked the question, are there differences in expression of KB4.3, K-chip 2, or KB4.2 in the ventricles? So these are Western blots again from the right ventricle, the apex, and the septum. There's no appreciable difference in K-chip expression or KB4.3 expression, but there's a steep gradient in KB4.2 expression, suggesting that KB4.2 is the primary determinant of this gradient, at least in the mouse. Okay, so what about the beta subunits? So this slide is just to show you that we know that there are multiple KB beta subunits expressed in the heart. There's KB beta 1.1, 1.2, and KB beta 2. There doesn't seem to be any KB beta 3 in the heart. And Ken Rhodes and his colleagues at Wyeth Ayres a couple of years ago described or developed a mouse in which they eliminated the KB beta 1 locus. And so we were interested to look at myocytes from those animals. And these are representative recordings from wild-type KB beta 1 knockout animals. So the beta 1 knockout will get rid of both beta 1.1 and 1.2, so both splice variants. And I think if you look at these records, you'll see that they look quite similar, but if you look at the details, the current waveforms are slightly different. And then when we analyze these records, what we find is that there is, contrary to our predictions, I should point out, there is a selective reduction in the ITO fast current and an augmentation of the delayed rectifier current. So if you remember, I postulated that because the beta subunits have been shown in heterologous expression systems and based on those immunoprecipitations from brain to be KB beta 1 specific subunits, these results suggest that the elimination of KB beta 1 results in the attenuation of the fast transient current and an augmentation of the delayed rectifier current, and no effect on the slow transient current in septum myocytes. And then if we do, yeah.
So what does beta 1 subunit combine with? Like can we which alpha subunit does beta 1 subunit?
So based on, so we've done two further types of experiments. One is immunoprecipitation to ask whether beta subunit co-immunoprecipitates with the KB4s. And the answer to that question is yes, and that's what's illustrated here. And then the other thing we've done is to now try to chop up the beta subunit, eliminating the N-terminus modifying the C-terminus and seeing whether these are multiple effects that are attributed to different domains of the beta subunit.
Jeanne,
Yeah.
following the logic you had before, what was your a priori assumption of what it was associated with?
We thought it would be associated with KB1 alpha subunit,
Right, which is what is supposed to be giving the fast transient.
the slow
Slow
transient.
transient.
Yeah.
So the data you got matches that?
No, it's exactly the opposite. So the physiology data
I see.
suggests we have an attenuation of the fast transient.
Right. That's assuming that the beta subunit enhances the slow subunit.
That's exactly right. So that's the interpretation of this result. But it doesn't prove that it's a direct interaction. I mean, there could be another protein here that's interacting with the beta subunit that's not the alpha subunit.
So those beta 1 subunits are also provide the ball and chain for the anti-inactivation, right?
That's exactly right. So you're exactly right. So when people have studied the beta 1 subunits co-assembling with KB1 alpha subunits that are not rapidly inactivating, the beta subunit confers rapid inactivation. So all of those sorts of experiments really suggested that it would be a KB1 specific accessory subunit. But when we do the immunoprecipitation experiments, what we find is that now we are precipitating with a KB beta 1.1 antibody. And we find that the beta 1.1 immunoprecipitates itself, but also coming down in this complex is beta 1.2, 4.2, and 4.3. But the KB15 subunit, and I don't have it on here, we also know the KB14 subunit, it doesn't come down with the beta subunits at all. And I should add one other thing here that's also interesting, that I'm not going to show you the data that shows this, but we've shown that this KB1.5 encodes part of the delayed rectifier current in these cells. It's the current we call IK slow 1. And I've already told you that KB1.4 encodes ITO slow. So that means that in the same myocytes, you have two members of the same family, the KB1 family, KB1.4 and 1.5, that do not co-assemble to make functional channels. So if you were to take both of those subunits and put them in a heterologous cell, they will combine and they will make functional channels. But these results in intact myocytes say that even though they're expressed in the same cells, they're not co-assembling. And the reason for that is probably that they need to be co-translated to be co-assembled. That shows you another important aspect of doing experiments in intact cells as opposed to doing them in expression systems. So the expression systems tell you what can happen, they don't tell you what does happen. Okay, so then what we went on to do then was to generate another line of mice in which only the N-terminal domain of the KB beta 1 is eliminated. And that's just the protein data. And in these cells, I think you can see here that the waveforms of the currents are now markedly different. There's a big attenuation of the peak, attenuation of the fast transient current. And then when we look at the cumulative data here, I think you can see that there's a large attenuation of the fast transient current, and now there's no effect on this delayed rectifier. So this then suggests that the N-terminal domain is important in this regulatory process, but not in this regulatory process. So we're currently doing experiments to probe the role of the C-terminus of the beta subunit in the regulation of the fast transient current. And I have to say, we're assuming these interactions are direct, but we haven't proven that. So it's important to emphasize that we have not shown yet that these are direct interactions. So it's possible that there are other proteins that are actually intermediates in this process. For example, patient. Okay, so then just to remind you what I think I've shown you is that each of the different types of channels that we characterize electrophysiologically are encoded by distinct alpha subunit genes. And now, in addition, when we start to look at accessory subunits, it looks very much like individual channels probably reflect the co-assembly of multiple accessory subunits with individual alpha subunits. And then from the work in lots of laboratories, and not ours, but lots of other laboratories, we could put together a picture that suggests that these channels are actually regulated and modulated by a host of other different factors. Both interactions with the actin cytoskeleton, this is something we're probing at the moment. We know that the KB4 alpha subunits interact directly with filament C, so we're actually beginning to explore the relationship between these actin binding proteins and the functioning of these channels. And then in addition, we know that these channels are regulated, and I'll show you a little bit I'll show you one example of this, by protein kinase mediated regulation. And then in addition, there's evidence from several laboratories that both voltage-gated sodium channels and other channels interact directly with the extracellular matrix. And one of the exciting things about this from our perspective, and I think this is true for other channels like connexins, gap junction channels, is that these sort of interactions begin to suggest a relationship between cardiac structure and channel function. So if we think about pathophysiology, where the extracellular matrix is altered, the channels might be altered directly as a result of those changes, rather than something through changes on the inside. And then finally, we've begun to explore the effect of changes in metabolism in the regulation of these currents, and I'll just give you a couple of examples. So in a collaborative study with Mark Anderson's lab at the University of Iowa, we've begun to look at the modulation of these currents by activation of CAM kinase, and specifically by inhibition of CAM kinase. And so Mark's laboratory has made mice expressing peptide inhibitors of the calcium calmodulin dependent protein kinase 2. And these are representative recordings from myocytes isolated from those animals. And interestingly, what we find in those animals is a selective upregulation of the fast transient current. And we've actually, I'm not going to show you the data, but we've gone on to look to see whether there are changes in message expression or changes in protein expression, and there aren't in this model. So this would suggest that there are changes now in the functional cell surface expression of these channels, or in the functioning of the channels, that are dependent on a post-translational modification of either the alpha subunit or the accessory subunit. And one of our interests now is to actually begin to look in this model and in other models directly for these post-translational modifications of these channels. And I should also add one other thing. This is chronic inhibition of the kinase. This is not acute inhibition. And so in the same way that I showed you that in the myocardium, when we knock out one channel with a dominant negative and get an upregulation of another one, that is a remodeling that you see in vivo that if you do a short-term experiment, you don't see. So it's possible that this is mediated by a mechanism that's not CAM kinase-dependent phosphorylation of channels. Yeah.
In Mark's Nature Medicine paper where they made infarcts in those mice, have you considered looking at IPO remodeling in structural heart disease in the inhibited mice and the normal mice?
Yeah, actually, we've just gone on recently to do that. Absolutely. And there are other channels that are remodeled in these mice. It's not just the
remodeling spots is different.
That's right. That's exactly right. And so, I mean, this is one of the interesting questions, and I think now in this system we can begin to tease apart these things that are long-term, short-term, and how those mechanisms are really different. I think they're going to be different.
Gee, are these channels actually themselves are modulated by CAM kinase or phosphorylated?
So there are two CAM kinase phosphorylation sites on KB4.2, and there's 3 on KB4.3. But we don't know yet whether they're phosphorylated endogenously or we don't have specific antibodies, and we literally have just begun to try to look at the protein chemistry to be able to ask those questions. There are sites we don't know if they're modulated. There's evidence from in the nervous system that the KB4-2 alpha subunit is modified by protein kinase C, protein kinase A, and CAM kinase 2 in hippocampal neurons directly. So one of the big things now that people would like to have is phospho-specific antibodies to each of those sites, and to be able to do proteomics to identify those sites. And that's, I think, sort of where the field is right now. And then finally, I just want to add that we also have begun to look at the effects of changes in carbohydrate and lipid metabolism on the functioning of these channels. And these experiments have been done in collaboration with Dan Kelly and Brian Fink and also Jean Schaefer. And I'll just show you one example of the sorts of things that we've been doing. So these are recordings, these are actually potential recordings from myocytes isolated from a transgenic mouse developed by Brian Fink, in which they've overexpressed the PPAR alpha transcription factor, driven also by the myosin heavy chain promoter. So again, you're getting robust expression of this transcription factor. It's known that the PPAR alpha regulates the transcription of a lot of genes involved in the regulation of fatty acid synthesis and metabolism. And so they have actually exploited this model as a model of diabetic cardiomyopathy. But because we are focusing on the most important channels, we wanted to know whether potassium channels were modified. And so what we did then was we recorded, again, isolated myocytes from these animals. And I haven't gone through it, but typically in all of these experiments, we exclusively record from cells from the apex, so we don't have to worry about the heterogeneity through the ventricular myocardium. And just focus your attention over here on this bar graph. What we find is an attenuation of the fast transient current in this model, and interestingly, an upregulation of this steady state current. And I didn't point it out, but I will now, and I think it's actually quite clear in these records, that although we've identified the molecular correlates of ITL fast and IK slow, we have not to date identified the molecular correlates of the steady state current. But we do know that it's not encoded by any of the KD subfamilies, because we made dominant negatives of all the subfamilies and made mice that express those dominant negatives, and we never touched this current. So we were, needless to say, quite excited about the fact that we not only affected this current, but we upregulated it. So whenever you upregulate something, you can leave it for
That's just the more important channels.
That's right, exactly. Exactly, that's where the comment is leading up to my punchline. So then we did a lot of pharmacological experiments trying to see whether we could figure out what this channel likely was. And I'm not going to belabor all the details here, but there were a couple of hints based on the fact that there was increased sensitivity to these relatively non-specific, but nevertheless somewhat selective potassium channel blockers. And this led us directly to a new type of potassium channel subunit that's been described in a whole host of different tissues, but primarily originally in epithelial cells. And these are, actually they were first identified in a nematode, what are called 2 pore domain potassium channel alpha subunits. So they have 4 transmembrane spanning domains and 2 potential pore domains that are transmembrane. And so it's speculated that these channels, instead of coming together as tetramers, come together as dimers. And so they are sort of like the inwardly rectifying potassium channels that you've all heard Colin talk about, and people in his laboratory study extensively, which they assemble as tetramers, but the 2 pore domain are like a dimer of these 2 channels. And so one of the things that we're focusing on right now, actually, in the RICO in my lab, is doing these experiments, is testing directly the hypothesis that the steady state current is encoded by these 2 pore domain potassium channel subunits. And then finally, I'll just show you another example of very rapid remodeling of potassium channel currents. And this we just focus on the left part of the slide. In wild-type mice, isolated from animals allowed to eat normal chow versus animals that are fasted, we see marked attenuation of potassium currents. And interestingly, the steady-state current that I mentioned is upregulated in the MHC PPR mice is not affected at all, and all the other currents are dominated.
Is this because of changes to the channels, or it could be because of potassium depletion, or changes in potassium concentration?
We don't know that. We don't know that. I mean, we don't know that. I mean, we don't have any reason to think that that's the case, but we haven't done any experiments that would really allow us to eliminate that. But it again points to the idea that you've got rapid remodeling versus long-term remodeling, and probably the molecular mechanisms underlying those processes are. Yeah.
You see all those reductions with the transient approach, you should see acupuncture prolongation in one and not the people on that.
Yes, and we haven't done that. But yes, that's something we need to do.
What are the half-lives of the LK channel?
So it's not known. I mean, nobody's done the elegant experiments, the counterparts of what's been done with the connexins. If you isolate myocytes and you use antisense on isolated myocytes, you can get effects on the order of 24 hours. So we think that it's rapid, but we don't know whether it's 4 hours or 14 hours. So, but there are lots of things that could be happening here. I mean, it could simply be trafficking. We don't know where these channels are. We don't know whether they're actually turned over or modified or, yes.
If I'm reading this correctly, we don't get a change in the fasted PPAR alpha question.
That's correct.
So if you treat the wild type with PPAR alpha, can you reproduce?
That's another experiment we haven't done, but that's something that we should do. Absolutely. Absolutely. But again, it might not be the same because this is a chronic overexpression of PPAR alpha, and it might not be the same as an acute overexpression. But we can do all those sorts of experiments.
But it's an acute effect of fasting.
That's correct. Absolutely. Absolutely.
I think you could say fasting changes the channel via PPAR alpha activation and the effects.
Yes, it's possible. It's possible, definitely. So, I mean, but what we can do all these sorts of experiments on isolated myocytes. I mean, these were done by fasting the animals. We haven't done it at the level of mice. So we could do that. And then the other experiment, which we've already been talking about doing, which is exposing isolated myocytes to alterate to different fatty acids in vitro and asking them to be direct conjugation. And then the other thing that we're trying to do actually is to put epitope tags onto this KB4 alpha subunit in a place that doesn't change its function, so that we'll then be able to put that into an animal and look directly at trafficking and recycling of the subunits of the channels in the tags.
Can you see the protein level change here?
We have looked in the transgenic animals for changes in protein, and we don't find them. We haven't looked in the fasting animals, but it's something that we may get into now that there are ever increasing number of people around here that know much more about lipid metabolism than I do. So I'll just end with this idea that I think all of these channels, not just our favorite channels, are functioning in this way, and that in order to understand them, it is necessary to study them in the intact environment, and it is necessary to sort of try to piece together this molecular regulation, but one subunit or one molecule at a time. And then finally, the most important thing is to acknowledge the people in my lab who've done all this work. A lot of the work I haven't talked about, which was founded on work by Diane and Sylvain, and Frank did a lot of the beta subunit work. And Noriko, as I mentioned, is doing the 2-4 domain work, and Celine is attempting she's looking at remodeling and attempting to do the sort of proteomic approaches that I mentioned. And she'll hopefully in the next year be able to give you a big update on that story. And then these are the technical people in my lab who take care of the mice and do a lot of the engineering of mice and analysis of mice. And then finally our collaborators here. I didn't mention the work that we've done with Jeanne, but Dan Kelly and Brian, the MHC, PBR mice, PBR mice. And then finally colleagues elsewhere that I'm sorry, it doesn't look like it shows up here. Barry London and Ken Rhodes and Jim Trimmer actually has been collaborated with for a number of years. He's responsible for making a lot of the potassium channel subunit antibodies that have allowed us to do a lot of these experiments. And then finally Tom Schwartz, who's made mice that I didn't talk about. And that's it.
Thank you very much. Questions?
So you mentioned in the beginning about another source of diversity is splicing variants. How do you actually look at that?
So we haven't really looked at those. It turns out that in vertebrates, splicing isn't that substantial compared to what it is in Drosophila. So in Drosophila, the main way to get diversity is splicing in the same transfer. So we know that KB4.3 there are two variants in the human heart. And we know that KB1.5, there are a couple different variants. For the others, we really don't know. But there are two reports out there that in human heart failure, there is a splice switch for the 4.3, and so it's one thing that it would be interesting to begin to look at what regulates splicing, but we haven't done that.
Also, there are reports about how to undo Subunits or that calcium channel.
Oh, absolutely. Yeah. So for the calcium channel, yes, for the calcium channel, there's extensive alternative splicing. So the alpha 1D and 1C, there are splice variants which are tissue specific and cell type specific, and then are regulated also by pathology. But yeah, we haven't done anything with those.
So I've seen there is already well-developed technology for human splicing variants. Something I believe is coming also for the mouse. So the mouse already technology. The same company, the same which made it for humans. So there's a mouse
Chip experiment.
What splicing?
Yeah, I'm sure there is actually.
So you can go ahead.
Yeah, we have not done that, but I'm sure there is.
For humans, I believe there are already 1,700 splicing variants in the chip available for insurance.
Yeah. Really? But is that mostly calcium channel?
No, all iron channels.
Oh, yeah.
We were actually sending some tissue from human avian ventricles versus apiary to try all these iron channels, splicing variants. And with a group which did work on human alpha and disability. The national stage.
Right. Yeah, absolutely. No, we haven't done anything.
Is it known whether the channels are in particular membranes or labs?
Yeah, so there are a couple of labs that have started to look at that, and it's only limited its success at the moment. So there are a couple of reports out there that KB1.5 is differentially distributed in caveoli compared to the KB4 subunits. But one of the difficulties is the tools that are used to do those experiments, and so we are severely limited in terms of antibodies. And most of the antibodies, I mean, this is a caution to anybody that tries to do these sorts of experiments, and we've come to, having learned by making mistakes, that if you don't have a knockout of the subunit that you're interested in, then you better worry about interpreting the results. Because most every antibody that you use in a Western blot recognizes several bands. Even if you can pick out the one you think is the one you're interested in, those other ones are still there. So if you then go to do immunohistochemistry, you're really at a loss to know what you see. So that's why we're trying to put epitope tags on the channels themselves, so that we'll be able to look at them directly. So that's sort of the approach that we've taken rather than the antibodies. But this is definitely what people are thinking is probably going on. So with sodium channels and calcium channels, there's a little bit more evidence that they're localized in caveoli and are just shuttled back and forth to the surface membrane from those. But here we don't. Yeah.
Did you look at those action potentials? What happened to the form of action potentials when you make changes of those channels, especially like in the case of fasting?
We haven't done the fasting, but we haven't done the fasting. I mean, we could do that, and this is the sort of thing we'd expect to see. So these are myocytes isolated from those double knockouts, if you will. And you can see that at the normal frequency of the mouse heart and at physiological temperature, you get a real prolongation of those action. So we'd expect to see exactly that.
Yeah. So you also show that the curves for humans and for mice are actually very similar. What is the major determinant makes the action potential look so different?
It's the relative densities of the currents.
Is there
So
any-
in mice and in rats, these transient outward currents are really expressed at very, very high levels. It's actually the main difference between atria and ventricle in humans. The fast transient current is what gives you that rapid early repolarization. So it's all it is the density. So but then you're going to ask the next question. I know you are, because you're a biophysicist, but is why then don't you get a plateau here, right? And that is, we don't know the answer to that. So it's either this steady state current, in which we would speculate that if we knock it out, we would get a big effect, or there's a contribution from sodium channel inactivation, like you have in axons. But we don't know the answer to that. We don't know. But we would love to be able to hit the steady state current alone and then together with these. And, if we could make a mouse have an action potential that went like that, it'd be great.
And that possible because of calcium channel.
Pardon?
And that possible because of calcium channel.
It could be because of calcium channels, but in fact, a number of years ago, we and other people have demonstrated that in rats and mice, the calcium channels are effectively indistinguishable from what they are in larger mammals. So it probably isn't a big contribution from the calcium.
But the question is whether, well, first of all, the density, and second, are they clustered the same way in two tubules, and all the whole story of ionic environment could be very different, and the channels would be different.
That's true, but if you just look at the macroscopic currents, they're not different in terms of density.
Yeah, but it's not enough to say that
That's
their
right.
contribution to the action potential is the same.
That's right, I agree.
But if you double calcium current with isoternal, you don't suddenly get a plateau of plus 20 in miles per hour.
No.
That's right.
Yeah, you do.
But if you change the properties of the kinetic properties of each channel, you could. Yeah, because you maintain a much greater late current, not affect the spike of the initial current.
So if you use barium, I've never done that. Have you done that, Tom?
No.
No, I've never done that. I don't know if you use barium as a charge carrier, whether you can make the action potential out of a plateau. I don't know the answer.
But if you knock out IK1, you still don't have a plateau, so...
Yeah, that's true. That's
Yeah.
right. That's an amazing story. If you eliminate the inward rectifier, you're still going to have it.
I meant to ask you, have you looked at those that are with the chem kinase inhibition, the Mark Anderson story? Are there any changes in the kinetics of those channels?
Of what, sir?
The chem kinase inhibition mouse.
Uh-huh.
Are there any changes in has anybody looked at the kinetic properties of the ion channels? Because you just showed MQT.
Yeah, for the potassium currents, there's no change.
No change.
And he's already published for the calcium current, there's no change.
Yeah, no, but the potassium channels there is.
No, there's no change in kinetic properties.
Any other questions? Thank you so much.
Okay.